So for the first part of today's lesson, we're going to look at Simpson's Index of Diversity, because we have just learned about the biodiversity, and we learned that there are 3 levels of studying biodiversity. So the first one is ecosystem diversity, second one is species diversity, third one is genetic diversity. So for Simpson's Index of Diversity, we are actually calculating the species diversity and then comparing this species diversity in 2 different areas or in 2 different communities. So students should be able to use Simpson's Index of Diversity to calculate the biodiversity of an area, in this case, yes, species diversity, and then to state the significance of different values of D for different locations. So higher D means what? Lower D means what? And then the formula will be given to you in the form of mathematical formula. So if you look at this diagram, which part has higher species diversity? By now you know already, right? Part B, right? Because there are 2 criteria for species diversity. So Simpson's Index of Diversity, once you have the data about the abundance of species, that means you go to these two parts, part A and part B, and then you go and collect data, take sample of the species in these 2 areas, how many species they have, each species, how many number they have, right? Then you can use this data that you have obtained from your sampling of these 2 areas to calculate the value for species diversity. So we use Simpson's Index of Diversity to calculate species diversity. So in Simpson's Index of Diversity, he also took into consideration that species diversity is governed by 2 factors. Number 1, species richness, means total number of different types of species. Number 2, species evenness, means each species got how many number of individuals. So it's abundance of each species, means total number of individuals in each species. So this is a formula that will be given to you. So where D is the index of diversity. Okay, this is Simpson's Index of Diversity. N is the total number of individuals in a species, small n. Big N is the total number of organisms of all species in the area that you sample. And the value of D must be between 0 and 1, whereby 0 means low species diversity and 1 means high species diversity. Okay. So for this Simpson's Index of Diversity, a lot of examples are given based on the community, means the different types of species that you can find in the rocky shore. Rocky shore means, you know, sometimes when you go to the seaside, right, it's instead of the very beautiful beach that you have, you can see a lot of rocks on, right? And then these rocks and a lot of waves will start to actually hit onto it daily. And sometimes high tide, sometimes low tide. So this is called rocky shore. And then there are all sorts of organisms, many different organisms here. So it's good for studying. Okay. So you can refer back to this picture when you come across the name of the organisms from the rocky shore later on. The more common one are the limpets, barnacle. Limpet are like a shell like that, okay, that stick onto the rock, okay. Barnacle is also like shell, but right in the center is actually like a hole, right? It's a shell, but in the center is a hole there. Litorina is like the snail. Then you got mussels, the one that we eat, you know, very common one, the mussels that we eat. And then this is called what? Starfish, okay, starfish. And then a lot of algae, a lot of seaweed, different kind of seaweed, sea anemone, sea anemone, this one here, okay. So example. So you can see that here is high tide zone. This is lowest high tide, middle tide, lowest tide. So you can see that this area, right, is where sometimes got water, sometimes don't have water, but they can still survive. Okay, and they can be exposed to sunlight. So this is example of rocky shore community. When it's low tide, you can actually see the starfish. You can see the barnacle, this one with the hole in the center. You can see the mussel, right? Example. And some seaweed. So a sample was made of the animal. Where is it? Animals living on 2 rocky shores. So shore A and shore B. 10 quadrats were placed on each shore. 10 quadrats means, you know, in an area that's very big, we don't go and count every individuals, right? We take samples. So they take 10 samples from the shore. Like if here, you'll learn the sampling tomorrow. But today I just illustrate that if this is the shore, let's say if this is the okay, this is a sea, yeah, this is a sea, and this is a shoreline. Okay, so they take 10 samples, means they make a fixed area. They decided a fixed area, probably 1 meter times 1 meter, and then they randomly put 10 of them, okay, in that area randomly, yeah, okay, and then they sample within this quadrat. So this, each of these boxes is called a quadrat. So they sample all the different species and number in this quadrat. So there are 10 quadrat in each shore. So this 10 quadrat will represent the species in this these 2 shores. Okay, representative sample. Okay, so shore A and shore B, 10 quadrats were placed on each shore, and the number of animals of each species in each quadrat was counted. The results are shown in the table. So these are from 10 quadrats. Okay, so to determine Simpson's index for shore A, first you must calculate n over n, isn't it? Okay, n for every species divided by n is the total number of organisms for all species that we have obtained from the 10 quadrats. And then square each value, add all of them up, and then subtract from 1, repeat the procedure for shore B. So we'll do this together. Okay, are we going to do it together? So this is for shore A. This is for shore A. So n, these are the result of the sampling. So after the result, we need to do the calculation, right? So just take the n divided by the total n here, okay, and then you square them. So after you square them, you add all of them up. So you get this value. Then Simpson's index of diversity will be 1 minus this value, 0.201. So that will be 0.799. Okay. So this one is for shore B. You will need to do for shore B. So can you do that for shore B? So for shore B, you have calculated that the Simpson's index of diversity is actually 0.733. Right? Can you get 0.733? Can? Okay. So now we have got the data already. So the D, which is index of diversity for shore A and shore B. So shore A is 0.799 and shore B is 0.733. So from the table, explain why shore A has a higher D value. So shore A has a higher D value, 0.799. So when you explain why it's got higher value, we need to know that the Simpson's index of diversity will take into consideration both species richness and species evenness. So we have to mention about this, both of them. Okay. So in terms of species richness, shore B is absent of 1 species. Snake, lox, anemone is absent. Shore B. You look at the table just now. So there's less 1 species than shore A. So shore A has got higher species diversity in terms of species richness. Then in terms of species evenness, we can see that shore B, the number of individuals of the species are lower, and beetlet anemone is also lower, but shore A has many higher number of individuals in many species, right? In all these species here. So therefore, shore A will have got higher species evenness because a lot of species are high in number, whereas for shore B, a lot of them are lower in number. Okay. So what is the advantage of this method is, it's not necessary for us to identify the species name. As long as you see, okay, this one looks very different, then you classify as species, 1 species. That one looks, even more different is another species. So you don't have to know species name, you can still find out the species diversity. Okay. But the disadvantage is that sometimes, right, some species, some organisms, they are of the same species, but they can look very different. And because of that, we might classify them into different species when we do the sampling. So that could be a mistake there, because the way we classify them. Okay. And we can only use the Simpson Index of Diversity to compare two areas when their species are similar in these two areas. So that means we can only compare like for like basis. That means all species in these two areas must be similar, then only we can compare these two areas in terms of species diversity. Okay, so we cannot compare species diversity of fish in the lake versus the moth in the forest, because different species. Okay, so that is the advantage and disadvantage of Simpson's Index of Diversity. So in this lesson, we have learned to use Simpson's Index of Diversity to calculate the biodiversity of an area and state the significance of different values of D. That means higher means what, lower means what, and then what causes it to be high or low. So that's all for this part of the lesson.